Jai Hind class. In the last class we have studied about acids and bases. Based on the pH of a solution or a substance, we can classify them into either acid, base or neutral. So, even we have studied how to identify a substance as an acid or base based on its taste. Edibles which taste sour are usually acidic in nature, and edibles which taste bitter, they are usually basic in nature. But we cannot taste every substance just to know whether it is an acid or a base, right? So here comes the word indicators, which will help us to know whether a substance is an acid or base. So that is today's topic, indicators. Let us see what are these indicators in detail. Indicators are nothing but chemical substances that are useful to test a given sample whether it is an acid or a base. How does these indicators work? These indicators changes their color when it is reacting with the solution. As the pH of the solution changes, the indicator color also changes. For example, any given indicator when reacts with acids will give one particular color, and when it reacts with the base it gives another color. So based upon that particular color we can decide whether the given solution is acid or the base. So there are types of indicators based on their sources. There are two types of indicators, natural indicators and artificial indicators. Natural indicators are nothing but the chemical substances which we can obtain from the natural resources, whereas artificial indicators are prepared in the laboratory by using different chemicals. That's why they are called as synthetic indicators. First we will learn about natural indicators in detail. So as we discussed, natural indicators are available naturally. Here we have taken three examples, mainly turmeric, China rose and litmus. So first we will discuss about the turmeric as a natural indicator. So you know that turmeric which is available in our kitchens, so it is a natural indicator which is easily available. So when it reacts with acids, it gives no color change, but when it reacts with the bases, it gives red color. Okay. Next example is the hibiscus, which we call it as China rose. It is prepared by keeping that petals of that flower in the warm water for some time, then remove that petals. The obtained solution now we will call it as China rose indicator or hibiscus indicator. This is we can use for testing a solution as acid or the base. If we add this indicator to acids, it will give magenta color. If it reacts with the basic solution, it will give a green color. Next example is the litmus. It is a natural dye which is extracted from the lichens. Okay, so this litmus indicator is available both in the form of solution or in the form of paper strips. The most usually used litmus indicator is litmus paper. So in our lab we have blue litmus paper and red litmus paper. So what is this blue litmus paper? Blue litmus paper, when it reacts with the acids, it turns into red color, whereas when it is placed in the neutral solution or basic solutions, it remains as such. There will be no color change. Coming to the red litmus paper. Red litmus paper reacts with the bases and turns into blue in color, but it does not show any color change when it is added to the acid or a neutral solution. So here are the other examples of natural indicators. So these flowers, hydrangea flowers, are naturally occurring pH indicators in nature. Depending upon the nature of the soil, the color of the flower changes. If this plant is growing in the alkaline soil, it gives pink color flowers. But when it is growing in the acidic soil, it gives blue color flowers. Right? Another example of natural indicator is red onions. This red onions juice, this is available in our kitchen. Okay, if this red onion juice is placed in the acidic solution, it gives red color, and when it is kept in the bases, it gives yellow solution. Right? Another one very good example for the natural indicator is red cabbage. Here you can see variety of range of colors it is giving when it is reacting with a wide range of pH. See, when it is reacting red cabbage solution, again when it is reacting with the strong acids like 2 to 3, it's giving pink and red color, whereas when it is coming to 7 and 8, a little violet color, and 9 and 10, it's giving little blue in color. And when it is reacting with the pH 11 substances, this red cabbage solution is turning into yellow in color. So we can see different range of color changes depending upon the pH. So the next category is the artificial indicators. As I mentioned, it is prepared in the laboratories. So we have two examples to be discussed. First one is the phenolphthalein and second one is the methyl orange. Phenolphthalein. Phenolphthalein is actually colorless indicator which we usually use in our laboratory. So when it reacts with the acids, it changes, no color change will be there. But when it reacts with the bases, it turns into pink color. So this is how we can identify whether the given solution is the acid or base. If it is acid, no color change. If it is a base, it will turns to pink. Next artificial indicator is methyl orange. This methyl orange, when it is placed in the acidic solution, it gives red color, and when it is placed in the basic solution, it gives yellow color. So these are the artificial indicators. Now we have one more indicator which needs to be discussed, that is universal indicator. A universal indicator is nothing but the pH indicator or pH paper, which is usually prepared by mixing different types of chemicals, so that by using that single pH strip we can know the pH of the given solution. Earlier, whatever the examples we have seen, like China rose indicator or the turmeric one or the red litmus or the blue litmus, okay, phenolphthalein or the methyl orange, we were able to know whether it is an acid or the base, but we are not able to say exactly, at least not approximate value of the pH. We are unable to say whether it is a pH 2 or pH 3 or pH 8. But by using this universal pH indicator, we can give approximate pH value. Okay, the given solution is of pH 8 or near to 8 or pH 7 or pH 5. Right? Why we are able to say this one? Because this yellow paper is made up of different types of indicators, chemical indicators. So each chemical indicator is able to identify one pH. So when we are using this yellow universal pH indicator strip, we will be able to identify the given solution pH, at least the approximate value, by comparing it with the scale here. pH scale will be given on this pH strips here. See here, the pH 1 is showing pink color, and 3 it's showing little orange, and yellow, brown, little greenish, and coming to the 12 and it's becoming blue. So based on that color we can give approximate pH value also by using the universal pH indicator. Right? I think everything is clear. So we will discuss about the neutralization and the application of this pH in the different uh, fields and in our daily life also in the next class. So, meet you in the next session. Jai Hind.